came to the doctor and said, well, we know you have FSHD, we cannot confirm it on a DNA level, and now we can actually, but it also supports this unifying disease mechanism that we proposed in 2010. Now, we don't work in isolation. We work with many people worldwide, and I'm listing here my closest collaborators. Robbie Terrell, a neurologist in Rochester, Stephen Tapscott in Seattle, a molecular biologist, the neurologist in Nijmegen, George Padberg and Basil van Hengelen, another neurologist in Nice, Sabrina Sacconi, and a biologist in Melbourne, Marnie Blewett. There are many other people with whom I collaborate, but these are really my closest collaborators, and we try to move the field forward. This is my current group in Leiden. You can see it's quite a big group, and it is mostly actually funded by external funds, so we're trying hard to get enough funds in to keep all these people working every day. And I'm actually blessed because it's a very, very dedicated group of people. So let's go a little bit into the genetics of this disease. Many of you, when you hear that you have the diagnosis of FSHD, and you get some booklets about this, then you read that in most families, FSHD is an autosomal dominant disorder with incomplete penetrance. And this holds true for FSHD1, the most common form of FSHD. And then, of course, what does that mean? What does autosomal dominant mean? And what does incomplete penetrance mean? Well, I'm trying to explain that in this small family tree that I'm showing here, father and a mother and 4 children. And the basic concept behind this is that we have 23 pairs of chromosomes. So we have 23 pairs of almost identical DNA sequences, right? So one pair of chromosome 1, both chromosomes are almost identical. On those chromosomes we have our genes. And you have to see your genes are like a kind of recipes for proteins. So there's the information hidden to make your protein. So basically for each protein you have 2 recipes because you have 2 identical copies of chromosomes. Now if one of those recipes is broken, and that already results in a disease, then we call it a dominant disease. If both copies are broken, then we call it a recessive disease. What happens is that one chromosome will be transmitted to the child from the father and one from the mother, right? So in this case, in this cartoon over here, what you see is a father who is affected because he has one gene that is broken. So it's a dominant disease because it's already penetrant in the Now the chances of transmitting this chromosome to his offspring is 50% because he will either give this chromosome to his child or the other chromosome. So that's basically why in FSHD the risk of having affected kid, if you're affected by yourself, is 50%. So here in this case, this child gets this chromosome from his father and gets the chromosome from his mother, and he gets the chromosome where the gene defect is located on. So he will be affected. This daughter over here will get this chromosome from her father, and that one from her mother. So she won't get the broken gene, so she's not affected. So that is an autosomal dominant transmission. Incomplete penetrance means, and many of you realize that in your families, if it's a familial FSHD, that some people may actually carry that broken gene, but still do not get FSHD in this case. And that's about 20%, which we call asymptomatic. So if you really closely look at them, they show some symptoms of the disease, but they are not severe enough to go to the doctor. So that's what we call with incomplete penetrance. You have the broken gene, but you're still not you're not disease presenting. So those are these 2 terms that I show here. Now I need to go a little bit back into the central dogma of biology. So we need to go back to, I think, middle school. Because this is essential to understand what's happening in this disease. So I told you our chromosomes. Our chromosomes is the genetic information that is the recipes for our proteins. And they are here in the nucleus. So this is a cell that contains 3 compartments, a nucleus. That's, let's say, the library, where all the information is stored. That's where your DNA is. That's where your genes are. And there is this cytoplasm, and then there is this wall, which we call a cell membrane. Now, this information needs to be faithfully copied to daughter cells, because every cell needs this information. This is what we call replication. That's basically just copying your DNA to be passed on to the daughter cell. So if there is a mistake in that, if there is a gene with a mistake in that, that mistake will be copied to the daughter cell. Now this is very precious information, these genes. Again, I would like to refer to this library. Just see it as a library with old books. You know, those books with those recipes, they are so precious, you do not want them to leave the library. So the cell doesn't want that information to leave the nucleus. So basically what it does, it has a lot of copy machines. So it just makes a copy of every gene. And that copy, or that copy of that recipe, you can bring home. So you can bring to the cytoplasm to bake your cake or to make your protein. So this is the central dogma of the biology. The genes are in the nucleus. They need to stay there. You need to be able to copy them to be passed on to the next cell. But if you want to use that information, you copy that information and you bring it outside your library to make your protein. This is not a simple task for a cell. And I always show this slide over here because each cell contains 2 meters or 6.5 feet of DNA. And you can compare that as taking a rope half the size of a marathon and trying to wrap that up in a tennis ball. That is what every cell is trying to do. So you can understand that's not an easy task. Now, fortunately, we do not need all our genes, because a muscle cell and a skin cell or a lung cell, they all have the same information. They have the same library, but they look very different from each other. So the cell only uses those recipes, those genes, that it really needs. And that we already know for many, many years, because if we take an electron microscopy photograph of a cell, this is again a nucleus, what we see here, so this is the library, then you see that there are black spots and white spots. Now these white spots, that's where the DNA is accessible, where you can read the genes, where you can read them, you can copy them, bring them home. But most of it is actually over here, in this very dense packaged part, which is black in here. And you can understand, this we can't copy, we can't have access to it. So only the cell makes available to, or the nucleus makes available to the cell what is necessary to maintain the integrity of the cell. Now, in 2010, I already referred to that, we had our breakthrough and we said FSHD is caused by inappropriate production of a DUX4 protein, dus inappropriate production of a recipe in the muscle of FSHD individuals. So what does that mean? Well, here I'm trying to explain the genetics of FSHD. We all have on chromosome 4 a repetitive DNA structure that we call D-vol-C-vol. And this repetitive structure is in non-FSHD individuals vary somewhere between 11 and 100 units. So it can vary a lot. And as a consequence of that, it is in this closed part of the genome of the chromosomes in our muscle cells. So if there is a recipe in there, and there is, it's compact, it cannot be read, it cannot be copied, cannot produce a protein. Most people with FSHD1 have a reduction in number of repeat units, leaving only 1 to 10. And that's the FSHD1. This is the most common form of FSHD, accounts for more than 95% of individuals. As a consequence of that reduction, this repeat moves to this open part of the spring. So now suddenly, if there's any recipe in there, it's available to the cell, to the muscle cell. Suddenly it can copy this and make a protein. And that's what the muscle is doing. It's making this protein which is called DUX4. and DUX4 is toxic to muscle. So basically what the muscle does in patients with FSHD is making a protein it should not make, and it is toxic to muscle. There is a second form of FSHD, which we call FSHD2. There we see the same change in location from this closed to this open part, but this is not due to a contraction, not due to a limitation of the number of repeats. So here, this is the main mechanism for FSHD1. So normally it's a long repeat, it's in this closed structure, cannot be copied, and in FSHD1, as a result of a contraction, it moves to this open part, and now suddenly it can be copied and can produce a protein that should not be produced in muscle. Now there is this second form of FSHD, which we call FSHD2, and there we see the same happening. It's opening up, but this is not due to a contraction of the repeat, but because we are losing a protein, which is necessary to keep that repeat over here. This protein is called SMCHD1. See it as a kind of putty or glue. It is necessary to keep this repeat structure over here. If you lose it, again, this repeat will move to this open part, and suddenly a protein can be produced from the repeat, which should not be produced in muscle. So this is the basic mechanisms for FSHD1 and FSHD2, but it doesn't explain as to why there's so much variation in the onset and the progression of this disease within families and between families. And I find it the most interesting part actually about this disease from a scientific point of view. These are actually pictures of two relatives from the first description of this disease in 1884 by Landouzy and de Jorine. And you can already see, these are nephews. They're about the same age, but this gentleman over here is much less severely affected than the gentleman over there. And these are familial cases. These are nephews. They have the same genetic mutation. How is that possible? And I'm not so interested in why some people are much more severely affected. I think it's particularly interesting why are some people not affected while they have the mutation. What is protecting them from getting the disease? Because that might actually give us clues into what kind of mechanisms we can think about for therapy. And I think one clue comes from our identification of this SMCHD1 gene, this putty. Because with the help of Sabrina Sacconi in Nice, we actually found these families over here. And I will try to explain to you what's happening here. So this gentleman over here has FSHD1, so has this contraction of the repeat. He has still a pretty long repeat, so he's not so severely affected. This is what typically the form that runs into families. So onset around 20, 25 years, slow progressive phenotype. But then suddenly he has a daughter who was already wheelchair bound by the age of 15. And we really didn't expect that in this family. Turns out that her mother has FSHD2, so a mutation in SMCHD1, the other cause for FSHD. And both diseases actually come together in this daughter over here. So she has FSHD1 and FSHD2. And it yields a much more severe phenotype, showing that these two mutations actually act synergistically. And I think it allows us to think about what happens in families with FSHD1 only. Because here we also see that many people are differently affected while they have exactly the same mutation. And what we think is happening is that the spring is to different degrees opened up between individuals. And that's what we call epigenetics. It's not the DNA sequence itself that's different between these individuals, but it's how we deal with it that is different between individuals. So we are starting to see this picture over here that in FSHD1 and FSHD2, because of the two different mutations, we see that the spring is opening up and the recipe can be read and be produced while it shouldn't be produced. Well, in FSHD1 and 2, so where you have both diseases, this spring is even further to open up so that much more recipes can be copied and can produce this protein that is toxic to cells. So how can we use this information now to think about therapy? And I want to make a disclaimer that although we are very excited about what we are seeing in the lab, that still is years from applying this in patients. But at least we're moving forward. Now, since we really truly believe that we have enough disease knowledge now, enough knowledge about this mechanism to start thinking about, and can we treat this? So I told you this whole mechanism that, okay, it changes in location so that it opens up more, and then suddenly a recipe is being copied, and that recipe is being produced, and that is toxic, and it can be toxic through multiple ways. It can do all kinds of things in your muscles that are not tolerated by the muscle. So if you now think about a therapy for this disease, you can actually start thinking about, well, perhaps we can intervene at this place over here. If it moves from this closed structure in the spring to this open structure, can we move it back? Can we basically close that spring again? Or if we cannot, can we prevent that copy to be made? Or if we cannot, can we prevent this protein to be made from the recipe? Or if not, can we then, that protein is there, but can we block some of his toxic functions in the muscle? And you can also understand the higher up in the hierarchy, the more effective it will be, because here we might be able to block this pathway here over here, but that doesn't help these pathways, which are still toxic too. So we like to be as high up as possible. And that brings me really to the question, okay, if we know that we're opening up the spring, if we relax it and make it available to be copied, is that a reversible process? Because this is happening during early development. It ends up in your muscle like this. So can we actually reverse this situation? That's not a trivial thing. So what we actually try to do now is say, okay, we know at least one protein that is necessary to keep the spring in that closed position. That is this protein here, SMCHD1, the FSHD2 protein. Yeah? So what happens if we bring in extra SMCHD1 in patient cells, in either patients with FSHD1 or in FSHD2? So we do this in the lab. We take muscle biopsies, make muscle cell cultures from that, and then we bring in extra SMCHD1. What happens? This is the only piece of real data from the lab that I'm going to show you today. But here, what you see here are bar diagrams of the amount of DUX4 that is being produced by the muscle. So here we see are your controls, and these are myotube cultures. So this is, well, let's say muscle in a dish. Right? This is control, so there's no DUX4. This is FSHD1, 5 cultures. See, there's a lot of DUX4 being produced in these muscles. That's what we know already. This is FSHD2, also a lot of DUX4 being produced. Now we're going to add SMCHD1 to these cultures. This is before we add, this is after we add. So yes, it is a reversible system, at least in a Petri dish. If we add SMCHD1 to muscle cell cultures, both of FSHD1 and of FSHD2 patients, we basically almost completely get rid of DUX4 production in these muscle cell cultures. So I'm actually hopeful that this is a way towards a therapy. And again, I don't want to give false hope here, and this is going to take years, but it tells us that the system is reversible. Something that has opened up can be closed again. And that's what we're trying to do in the future. So this is my part, but I want to spend 2 more minutes or 3 more minutes on an initiative that I really like. And I promised Newcastle to tell something about it, and that's the UK FSH patient registry. And I cannot underscore how important this is. We have seen with many other rare diseases that we come up with molecules, and we want to test them in trials in patients, and we don't know where our patients are. And we have no registries about them. We don't know where they are, we don't know how they feel, we know nothing about them. And this has delayed in many other diseases the path towards a therapy. So I cannot stress enough how important this is if we want to make this transition from diagnosis to treatment. So Libby, she's also here in the room, and she can tell much more about it. But I'm very happy with this initiative, and I'm also very happy that many other countries are actually following Newcastle here in making similar registries so that we will have sufficient information about our patients in the event that at a certain point we actually come towards trials. So what is the kind of information? And Libby can tell much more about it than me, but I just want to show you a little bit. So you're asked about some personal data, clinical diagnosis, your motor function, wheelchair use, pulmonary function. Well, it's all listed over here, also whether there is a genetic diagnosis being set. And it has been a very successful registry already, because up to now, and your year for life now, less than a year? 2 years. 2 years. Already 560 people have registered. But my math said that there must be at least 5,000 patients in the UK. So we only have 10% right now. So, yeah, because we know the prevalence of the disease. So there's still a lot to do, but this is a great start, and I really applaud you with this achievement. Okay, so take-home messages. There are 2 forms of FSHD, FSHD1 and FSHD2. The common form is this contraction of this repetitive DNA structure. The rare form is most often caused by these mutations in this protein that is necessary to keep this repeat structure in this closed environment. Both form, and I think that's very important, can be genetically confirmed with very great accuracy. There's no need that people here do not get a proper DNA diagnosis for FSHD. Both forms, is also important, have an identical disease mechanism. They converge at this point where this protein is being produced because the repeat structure moves to this open part of the spring. So if we find a medicine for one, we probably also have one for the other disease. Some individuals have FSHD1 and FSHD2. This leads to a much more severe course of the disease, much more early onset, much more rapid progression. But it also tells us how we need to think about this variation within families and between families. So I think we have uncovered the mechanistic basis of FSHD sufficiently enough to move towards translational phase to discover therapies for this. And I think this can only be done with the help of you, because every day we use your DNA, we use your cells, we use your information to do these things. So I thank you for doing that and for your attention.
Brilliant presentation and explaining the mechanism and very much as well the basis. And I think we have now a lot of time for questions. And maybe can I start? Because I just have a question with people having 2 mutations. I mean, I would have, because FSHD itself, FSHD2 and 1 itself, are relatively rare.
Yeah.
So I'm quite surprised that in the more severe forms, you get like a combination of both mutations. I would have then kind of thought that each form would be much more frequent. So is it then underdiagnosed, or?
Yeah. So there's something interesting going on with this repeat, and I think that explains why we actually find these families, because I agree with you, they're so rare that getting them together, being combined, is almost, well, if you do that, impossible. But what we also know, so I told you that we have this contraction between 1 and 10 units, but about 1% of our population has a repeat of 8, 9, or 10 units. So is theoretically at risk of developing FSHD, yet they don't. And I think that explains why we find FSHD1 and FSHD2, because 1% of our FSHD2 population will also have FSHD1, a contracted repeat of 8, 9, or 10 units. And it tells us that we are basically in the 8, 9, and 10 units in this gray zone where bringing this repeat from the closed part to the open part might be an almost stochastic event. In some people it will move to the open part of the spring, while in others it will remain in the closed part. And that is in part due with what we call epigenetics, is, for example, by the efficiency of this SMCHD1 protein, which will be not as efficient in every person. So this is why I think we find these two diseases together.
And have you looked, I mean, I know that in a very kind of small number of people, you do get the very severe juvenile form. Have you looked in particular in those cases then?
No.
Is the juvenile form is different?
The juvenile form is in that, so what we see is a, very roughly speaking, a repeat size dependent severity. So those individuals with 1, 2, or 3 units, those are the infantile cases most often, so they're the most severely affected. And if you go to longer repeats, disease gets into the familial forms of the disease with the typical onset in the 2nd decade and the variable progression of the disease. And one factor that determines this variability is SMCHD1. But if it's these 1, 2, or 3 units, it is the most severe case. These are the most severe cases, and there the repeat gets into the most open part of your spring, let's say.
Good. So I'm sure there are questions. Yes, please.
I have FSH, but I've never been told whether it's 1 or 2. Not that I'm worried, but I've just got it anyway. But you may be interested to know that it's been in my family for over 100 years, and I am the 3rd generation. My maternal grandmother was a carrier and a sufferer, and so was my mother, and she carried it to me and my sister. My aunt was a carrier, but not a sufferer, and she carried it to one of her 3 children.
Yeah, that is a typical, I would almost say a typical family with FSHD, where you see these kind of segregation of the disease through the family. So yes, you see individuals who are carrying the mutation, but will never become manifest. But since it is in their DNA, they can actually pass on the mutation to the next generation. And that individual may become affected again, yeah. So this is something that we hear. So 20% of the gene carriers, 20% of the individuals who do have the mutation will never, be manifest.
I'm willing to help with any research.
Oh.
Yeah. Wonderful.
Have you been registered then? Have you registered yourself?
Yes, I think I saw my little blue dot on the map.
Good, very good.
and my sister.
Brilliant. Yes.
So I'm Christine, hi. So with genetic testing, does it actually show if you are a carrier as well as have the disorder?
Yes, well, they can, what genetic testing can do for you is tell you whether you have the mutation,
Okay.
whether you are showing symptoms or not.
Okay.
Yeah? So,
So it's possible to have a negative FSH genetic test but still be a carrier?
no.
No.
The reason we're asking is that both me and my husband have been genetically tested and we don't have the disorder, but my son has FSH.
Yeah,
So the geneticist said to me that in some cases it, I guess, just starts.
it starts,
out.
yeah.
Yeah. So what is the?
A family, I've only been talking about the familial form of FSHD, but it has to start somewhere, right? So sometimes when I told you this mechanism, I can go back a little bit. Oh, yeah.
Okay.
Here, this mechanism.
Yeah.
So I told you that if a cell wants to produce a daughter cell, it needs to basically copy all their DNA and pass that on to the daughter cell. And this is a very faithful mechanism. You have to realize there are 3.2 billion letters to be copied. Yeah, that's amazing. Yeah. So, and it does that extremely accurate. It almost makes no mistakes. But sometimes it makes a mistake. And most often that's not harmful at all. But if this mistake is by accident that you get a repeat contraction or a mutation in SMCHD1, then the child will have a new mutation. And that is in about 20% of FSHD1. So about 20% of FSHD1 individuals are because of a new mutation of the repeat. Yeah? So that also means that your chances of having a second child with FSHD are not greater than the average population, of course.
Yeah, because the other two have.
So, so for I'm registered with the FSH registries here and in the US, but I had my DNA test done about 16 or 17 years ago. They can't find them now. But I remember that at the time when they saw it, they said I had a rather large repeat. For me, my symptoms started showing when I was probably about 11, so it's pretty early on. Should I have another test done, or should I try to locate?
I refer you to Libby for this, because she knows all about the registry and the importance of knowing the DNA diagnosis.
Yeah, so I can't give you any advice in terms of the American registry, but in terms of the UK registry, we really would like to have your genetic confirmation, and you can be tested in the UK without, you should be able to at least. And when it comes to trials and it comes to studies, people are unlikely to be enrolled if they don't have a genetic confirmation. So yes is the short answer.
And it might also, and as I said, I mean, this registry, as both of you pointed out, is really, really important to prepare for clinical trials, because also it's a resource. It's a resource for pharmaceutical companies to do so-called feasibility studies. You know? So they as pointed out, they need to know where the patients are. But also maybe what might be is that a pharmaceutical company has some form of drug or treatment where they only want a certain subpopulation. So maybe they want people between a certain age, they want people who have a certain repeat, the deletion of certain number of repeats, and as such, it's really important to have your genetic data in there, because it's more accurate than is, as better the registry will work, and as more kind of we know about patients, and as more as well we can attract funding from the pharmaceutical industry. Because to be quite honest, I don't know, I think, hope you agree with me, but to develop a drug and to bring it to the market, more importantly, I think at some point you always need to have a pharmaceutical company involved. And then I remember, this is not a very long time ago, in Germany, that they tried to do a clinical trial at the very early ages for patients with Duchenne muscular dystrophy. They wanted to test the steroids, and it took them 3 years to recruit sufficient patients into the clinical trial. No pharmaceutical company will invest money into that area if that is the case, because it's just taking far too long. And that's also, of course, for you a lost time, because in three years muscle will deteriorate. Yeah, you have another question?
Follow question.
Sorry.
If it shows when it's earlier, does that generally mean that there is some familial involvement, or can it still be spontaneous?
I can only talk in general rules, because every family is different, and especially in FSHD, is that the case. But if I would say that if it's a very early onset, an infantile onset as we go, then most often these are the individuals with 1, 2, or 3 repeat units. And these are most often, but again, it's all generalized, new cases, new mutations, and non-familial cases. Yeah?
I just wasn't sure, because no one in my family's been tested. I think my brother is a little nervous about
since there's no cure, why, you know, he doesn't want to know if it's coming and whether or not his daughter has it. And I can certainly understand that. So I don't know if, you know, I should press for them to get tested or not in terms of that.
You know, I always tell, I think these are personal decisions. You know, I'm not going to tell people what to do here. I can only say, I think, if you want to be involved in the research, if you want to move on with the translation, then I would say, you know, get tested and get registered. But these are individual choices, and I respect them.
I mean, the only thing, yes, of course, I completely agree. It's an absolute personal, very, very personal choice whether you want to get tested or not. I think the only thing what one should maybe remember is that if you want, or if your brother, for example, you said he has a daughter, if he wanted to have further children, you can now use a technique that is called pre-implantation diagnostic, genetics diagnostics. So which is an in vitro fertilization, which involves in vitro fertilization, and they can then test an embryo, whether an embryo in the petri dish is affected or not. And as such, they can then choose an embryo that is not affected. But I mean, some people don't want to do that, and absolutely, I mean, it's a very personal choice.
Yes.
Some people feel there are ethical issues compared to them. Other people feel, I don't want to have another child with FSH. I have one already. I want to prevent it. And I think if you ask all these questions, do I want to know or don't I want to know? I think it's always good to make an informed decision and to know about all the consequences and options. So I think this is always what I kind of say as well. I think we had a question here.
I think it's also been in my family for over 100 years, but it looks as though in my case that it's developing at a later age with each successive generation. Is that likely to continue?
It's a very interesting phenomenon that you describe. It's called anticipation. And we know anticipation going in the opposite direction in another muscular dystrophy. It's called myotonic dystrophy, where you see that in further generations the onset is earlier and the disease is more progressive. And we have a genetic explanation for that. In FSHD, we have been looking into this anticipation, and I don't think that there is very good evidence that anticipation is happening in FSHD. But mostly, I have to say, we focused on the opposite, so that in every new generation, the onset was earlier and the disease course was more severe. So I think this is the first time I hear about this, that the onset is later every time, and I have no explanation for this.
I gathered that it was normally the other way around. But, I mean, my father was diagnosed with muscular dystrophy when he was 44. I was 70 when I got the diagnosis, and my paternal grandfather died aged 36. I think he was the start of it, because my father's sister also had it. But whether he died from FSH, death said pneumonia.
Yeah, no, well.
That was 1902.
Yeah, so I think the healthcare situation may contribute to some of these aspects, but in general, if we look in these families, then we don't see any evidence for that in next generations it's getting either worse or milder.
Mine is milder than my father.
Yeah, mine is milder and seems, this might not be the right phrase, dying out, but it's definitely milder as the generations have gone on in my family.
Very interesting.
Are there any more questions? Yeah?
Are there any other diseases that DUX4 is implicated in besides FSH?
The only thing that we know is from, this is called a translocation. Sometimes, I told you have 2 pairs of chromosomes of each. Sometimes not. There's, things go wrong, and then a piece of one chromosome is fused to another piece of another chromosome. This happens. I think that we have 10, 20 cases described worldwide, so it doesn't happen often, but it may happen, and then you get a fusion of 2 recipes. So you basically fuse the 2 things, and that may lead to a form of cancer in the muscle. But that's so rare, but it has been described. It's the only thing that I can think of where DUX4 has been implicated in. And then it's not a full DUX4, but a half DUX4 fused to something else. Yeah?
I maybe have a quick question. At the end of your talk, you had where you talk about treatments here.
Yes.
Yeah. So I don't know whether you want to maybe go into it, so that there are approaches maybe to inactivate maybe DUX4 at the RNA level, using antisense oligonucleotides or using drugs.
So that's here at this level.
Yeah. Which of your opinion? I mean, do you want to kind of maybe comment on that? How far is antisense oligonucleotides?
Okay, so again, I'm not a pharmacologist, so I'm on thin ice here. But what I hear from pharmacologists, the smaller the better. Yeah? The smaller the molecule, the better. And believe it or not, but an antisense is a big molecule. So you rather want to go smaller. Now, there have been some papers already, again, this is in petri dishes, this is in muscle cell cultures, that yes, you can design antisense oligonucleotides to this product here, this recipe, and that blocks the production of that protein. So theoretically, or in cell culture, it is possible. Is it the most promising strategy? I don't think I can say that. I prefer to go for what we call a small molecule, so a chemical modification, rather than an antisense. Having said that, you can also use antisense for this step, to put it back together, basically. And there are also publications have already been showing that this is, in a petri dish again, possible. There is one thing that concerns, what I think is a concern related to this, and that is the way DUX4 is being expressed in the muscle. And this is, I didn't bring a picture of this, but it's called what we call bursts of expression, right? And that means, so not that every cell is producing the DUX4 protein, but only a few out of many. But those few out of many that do produce a whole lot. So if you think about the therapy that basically tries to destroy all these products, then you need, in this one particular nucleus, in this one particular cell that is producing, and it's only one out of many, you need a whole lot of these antisense oligos. So I think it's a challenging concept, but people are working on it, and it doesn't, and there are some good data there. So I think at this point we should just focus. I used to have a boss who says we are focusing on everything, but I think it's a good thing. You know, we need to work on this and this and this and this, and we need to work on all of these possibilities, because at this point we don't know which of those is going to be the winner. So,
what do you think about? I mean, so you were pointing out that if you wanted to somehow basically close the DNA structure again, you need to kind of target as many of these nuclei in a muscle cell as possible, because if you only target a few, you still get DUX4 expression from the other ones, and you still get an effect. So, I mean, I know this would be a very long way off, but what about, there's a lot of talking about genome editing and CRISPR-9 technology at the moment that for me always kind of thinks, this is a technology where you would really repair the genetic defect, and combining that there is a lot of talk with stem cells, combining stem cells.
Yeah.
So what do you think about that?
Also,
I mean, it will be difficult to do it.
also people working on the gene editing,
Okay.
it's going to be very difficult in a sense that, so normally gene editing is done on a gene. And that means that there's one copy, or two copies because you have two chromosomes, but it's what we call a single locus. Yeah? It's one place. Here, in this individual, there are already 3. You know, in this individual, there are tens. So it's going to be more difficult to edit all of them. So I'm not saying that's not possible, but it's a little bit more difficult. On the other hand, turning this into that again, these are epigenetic drugs. And pharmacy has already produced a lot of epigenetic drugs for cancer therapies, and they're successfully being used. Unfortunately, they all do the opposite. They're opening up the things rather than closing it down, because that's basically the strategy that you need for some types of cancer. But it tells you that it is possible. It is possible to move between these two states. And if they can find things that open the structure up, then they must also be able to find things that close it again. So.
Do you find, and I'm sorry if I kind of didn't pay attention to, so is it the case that as much as SMSMUCH, SMCD is around, as more closed this structure is there a kind of quantity effect of?
Well, the only experiments we have right now is that if you increase the levels by twofold, you basically shut down DUX4. So you need a relative. Twofold is not that much, and we haven't tested 1.5 or 1.4 or 1.2. And what we also know is that if we look at the levels between individuals, they may actually, so you and I may actually differ a factor 8 in the levels of SMCHD1. This is apparently some natural variation that is allowed, that is tolerated. So, moving something from 1 to 1.2 or 1 or 2 maybe, it's not that much. It's not like 10 or 20 or 100-fold more.
So would you looking then at drugs as well, like similar what Kay Davis is developing now for DMD with utrophin upregulation? Are you looking then for drugs that can raise the level of?
Yeah, so we're looking in different ways, but the trouble is we don't know what SMCHD1 is doing. We know that you need it to keep it there in that closed environment, but what it's exactly doing, we don't know. So we're looking into this protein, and we're looking into ways whether we can increase either the levels or its unknown activity. And at that point, of course we would like to know what the activity exactly is, but as I said, we know enough about the mechanism that I don't want to wait until we know the exact mechanism before we start thinking about, can we increase the activity? So we're doing all kinds of screens at this moment to see, can we either increase the levels of SMCHD1 or can we increase its activity, whatever that may mean. And you do that basically by looking at DUX4 again. Can we find conditions, can we find ways that there is less DUX4 in the muscle?
Are there any clinical trials going on in the UK for
Is the UK involved in the ATIA trial? I don't know.
No, not that I know.
No.
What's the ATIA trial?
That is, again, has nothing to do with this part, but this is an immunomodulating agent developed by a company called ATIA. And because there's an inflammatory component to this disease, they are currently conducting a trial with this, and that's ongoing. And I know it's an international trial. I know people at least from the Netherlands and from Italy have been recruited, and I don't know about the UK basically. No.
Not in the UK at the moment.
Okay, thank you.
And I think if it would be in the UK, then it would be most likely that people who are eligible for the clinical trial would be contacted through the registry.
Oh, yeah.
Yeah. I mean, I just wanted to point out just one thing as well with the registry. If you're interested in research and if you want to know what's ongoing, then that's also kind of a good way to keep up with the scientific world, because we are writing, or Libby, we do that in collaboration. We are writing, how is it now, twice a year, or about twice a year, we are sending out a newsletter from the registry, and we normally work very well together to update everybody who is involved on what's happening in the scientific world. So if there then would be a clinical trial happening, that would certainly be as well in the newsletter.
I go to the local FSHD that in my area, and this is the first time I've ever heard of the registry. So it would be a good idea to kind of because there's quite a large number that attend when I go. But
Yeah,
I will let them know.
Yes, please.
I've got some leaflets there.
Okay, great. Oh yeah, thank you.
And also maybe what we should do from the charity, send out an email to everybody. I mean, we have done this, when we kind of do condition-specific emails, then we send them out to everybody whom we have on our database, in order to kind of ensure that everybody knows about it. So, has anybody got?
Just one question. Obviously, you're working very hard in the Netherlands. Is it similar in the States and France, and obviously here in the UK? Are you all working closely together? Obviously, you mentioned things here that obviously it's all double Dutch to me, but I've understood a lot more about it today. And excuse the double Dutch. I didn't know that.
I'm used to those things, Dutch streets. I know all of.
Sincerely apologise, but just seeing all those names there, and obviously.
So these are my closest collaborators,
Right,
but there are many other people in the US, in Europe, and in Asia working on FSHD. And also I'm very happy that also in the UK, it has been a while, but now the Newcastle is really catching up, and I'm very happy with that. And also Pete Samet is doing very good work here. And Jane Hewitt,
Jane Hewitt, who kind of really contributed as well to the understanding mechanism as well, because she always claimed that there was a gene here.
she was the first to describe
She
it.
was the first to describe.
Thank you.
So, yes, just one question. I don't
No,
know
it's not a question, it's just a thank you for the clearest explanation I've ever heard about FSH and what's here, how it occurs.
So one thing I can thank you very much.